Okay, I think we can kick off. So welcome everyone. Oh, yeah, go for it. Yeah, sure. Welcome everyone. We're at lecture 6. We could pull the door through a little bit. Okay. Lecture 6, Intro to Mathematical Neuroscience, back in the fields. And now we're going to talk for a bit about oscillations and synchrony. So the kind of style and the content for today is a little bit more kind of biology oriented than math oriented for the most part, this time round. But we'll pick up on a few of the themes that we've covered and that have come up in the last few lectures. So there'll be some familiar points, and we've got some, as usual, some excellent practical tutorials for you in the second half that Parza will lead. So this was our syllabus. We had a little bit of a jiggle around in the middle there. And we are maybe thinking of modifying the content of lecture 9, just in case people are kind of comparing this to the slides from a few weeks ago. There's some kind of minor adjustments, but the general structure is basically the same. So we sort of finished our kind of journey up through the spatial scales that we were doing in the first half, right? That was part of the theme for the first few lectures. And to some extent, the next few, this lecture and the next few, so oscillations, synchrony, noise, etc., are they kind of span all scales. Like, so the principles apply in, to some extent, individual neurons, small neural circuits, large neural circuits, macro scale, brain networks. We see a lot of these phenomena like stochasticity and oscillatory activity, like all over the brain, all over the spatial and temporal scales. So will be this applies. It is generally applicable. However, broadly speaking, we're kind of in this territory for this lecture. I mean, not broadly speaking, like we are definitely in this territory for this lecture. And what I mean by that will come through in a few slides. So there's, as I mentioned, so there's oscillations in the brain at multiple spatial temporal scales. You know, the ways which what we mean when we're talking about things at multiple scales is typically kind of related to the ways we measure them. So the certain types of measurement modalities that allow us to see oscillations in, say, patches of membrane or populations of neurons, or it's a small patch of a living person's brain during surgery, etc. And, you know, to some extent, like what we see, there's it's a bit of a filter, right? So we can see what we see in data is kind of mediated a little bit by what the measurement tools that we have allow us to see. We don't really have a kind of fully omnipotent view of the full range of oscillatory activity kind of across all the spatial scales. We have certain, say, temporal and spatial kind of frequency characteristics to our measurement techniques. And what that boils down to, or one of the ways that kind of manifests, is that, for example, and we'll kind of come to this, so we tend to see more high frequency stuff at smaller spatial scales and more low frequency stuff at larger spatial scales, as a rule. So we have rhythms all over the place. I mean, we have rhythms in other parts of the body as well. General question is why. in neuroscience, and particularly kind of human neuroscience, neuroimaging, the kind of origin story for a lot of what we do and a large chunk of what we kind of know is goes to this fellow Hans Berger, who was active in the early part of the 20th century. Credited with two things in particular, both first basically inventing or discovering the EEG, electroencephalogram, so measuring oscillatory activity on the scalp. And within that, characterizing its primary features, one of which is the main one of which is called the alpha rhythm, and originally was called the Berger rhythm, was named after Berger. If you have the time and the inclination to do a bit of kind of further reading on Berger, it's interesting. It's a little bit sad in a way. It's kind of life story, but it was inspired by things like a sense of having some telepathic communication with his sister before his sister died in an accident, and lots of weird stuff. But this, in any case, drove him to do some original science ultimately, although he didn't really see the benefits of it in his lifetime. But yeah, so the endpoint of this is, one of the main things is this thing we call the 10 hertz alpha rhythm. Okay, actually, Berger kind of gave us at least the initial structure that we use for defining the names of different brain rhythms in terms of their frequency ranges, as in like alpha, beta, gamma. He didn't, I don't think he gave us gamma. I think gamma came in the 80s. I'm not sure when theta came in, but I think alpha and beta were named as such by Berger, and then some other people followed the nomenclature over the subsequent decades. And then where we are now, particularly in the last 10 years, speaking personally, I think we've got a bit of a problem with the fact that we've kind of inherited the definition of frequency bands and thinking about things in terms of frequency bands like too onerously. But that's a bit of a digression. Broadly speaking, one of the key early findings was this 10 hertz rhythm that we see on the scalp, the alpha rhythm. It has variation in a number of important kind of cognitive states. Sleep is a very, very obvious and kind of universal one. Anesthesia, cognitive things like behavior, attention, kind of attentional focus, and pathology, and also aspects of neural coding. So that's kind of oscillations, and then a topic that kind of goes hand in hand really when we think about oscillations, particularly oscillations in networks, is synchrony and coupling and the tendency of independent units to group together. And the general question in neurobiology, like a kind of eternal question in a way in neurobiology, is like what role and what implication can synchrony in brain networks have for cognitive function, and like where's the causality? So like, do we see synchrony that's kind of an active process of, say, the neural code, or is it a kind of byproduct of neurons firing, and then it just kind of drives oscillatory activity? So this question of like epiphenomena that is still a live question to some extent. So, if we see like a rhythm at a network level or at a kind of measurement level, like an LFP or a scale PEG, or a rhythmic signal within some electrical field, to what extent is that just something that's a side effect of the neural activities in that part of the brain, and has no kind of functional implication at all? This is there's lots of ways in which this question comes up. We'll touch on one or two of them, like communication through coherence. There's other ones like aphatic coupling that we won't touch on today, but are related. But the bottom line here is like we're basically not too sure on a number of those questions around kind of causality in measured oscillations. Where we're at really is trying to kind of characterize them in terms of their dynamics and in terms of what how they seem to behave. So one of the workhorse, like say, concepts, mathematical models that has been used in computational neuroscience and in like a variety of fields to just understand coupled oscillators is the Kuramoto model. Okay, so this, I've got one slide on this, and then we've got some more discussion on this in the tutorial. So we're going to go into this in too much detail, but the general idea, Kuramoto from Winfrey earlier, is kind of define the most pared down, mathematically simple description of a system of coupled oscillators, and what can we learn from that general structure. So the canonical form of the model is that we have a set of units, oscillatory units that are denoted I. Okay, so we have n oscillatory units. Each unit is defined by its phase, and that is the, I guess, distinctive feature of the Kuramoto model. Is like we're not really talking about something like an amplitude signal in the way that you might think of an LFP or like a membrane potential or firing rate. We're talking about, where is it on its circular journey from 0 through pi and back again, right? So we've got a system that's defined in terms of the derivative of the phase, the time derivative of the phase, and the way in which our units interact is by what we call a difference coupling term. So that is the basically influence, the velocity of the phase is determined by the natural frequency of that oscillator. So each of our oscillators has some natural frequency, omega. There's going to be a distribution of omegas across the system or the network, if you like, which could be a tight distribution or a narrow distribution. And that's one of the things that characterizes the behavior of these systems. So, but each unit, each I has its own frequency, and then it has some input from all of the other nodes in the network, okay, scaled by the number of nodes, and some term that will parameterize in terms of like what the scale weighting would be, this k here. So sum and then sign on the phase difference, right? So this is basically saying if your phase is similar to mine, other node in the network, then we're great. Like that, there's going to be a kind of low term there. But if we have a big difference, then we get a push away. So what we kind of end up with here is a kind of pushing on the, we think about from 0 to pi on the phase difference. We're kind of pushing the difference coupling term here is doing for every node pair in the network, so a little kind of incremental addition here. We kind of push towards the kind of 0 or the pi. Okay, so the nature of the Kuramoto model is like it kind of allows the system to move towards synchrony, or the system tends to move towards synchrony, as in like 0 phase differences, and it's determined by these parameters that essentially give you the kind of coupling strength in the network. So that's the general setup, and then story of the Kuromoto model in computational neuroscience is kind of variance on the theme, but the variants are along the lines of what is the statistics of the distribution of the frequencies, natural frequencies of the nodes in the network. What is their network structure? Okay, so this setup is basically just saying it's a uniform structure. A kind of slightly extended version of this would be to say we have some weights matrix, right? And then so we might index our IJ weight a lot when we're doing this coupling difference, this coupling term here. We might use other coupling functions that may be a little bit more neurobiologically consistent with how we think about neural responses to inputs. And there's a long way you can go in terms of extent add delays. Okay, but they're kind of variants on a theme. Again, I think the key thing about Kuromoto model is the fact that you're describing the evolution of the phase rather than some kind of membrane potential activity variable. And then the thing that's the kind of key phenomenology of Kuromoto model is its behavior with respect to this quantity called the order parameter, or the Kuromoto order parameter. So this is a kind of simple scalar parameter that's defined by, or scalar, I say auxiliary variable that's defined by the parameter that's specified in the model. So this K parameter, and what happens is as you change the values of K, so for kind of increasing levels of coupling within the network, essentially, then you get a kind of rapid transition between a asynchronous state, or kind of random distribution of phases on the unit circle, and a highly synchronous state where you get a low amount of phase difference, which look a little bit like this, where we have a lot of clustering of the phases close to some point, some angle. Doesn't need to be there, it could be anywhere around the circle. But whatever the phase difference is that the system wants to kind of settle on for the specific parameters you set up, if the distribution of phases is narrow, then that's kind of defined as high synchrony. And that's something that is we kind of quantify by the Kuromoto order parameter. So I have a bit more on this later, but Kuromoto model, again, sort of the most simple type of oscillatory network model. And it gives us a lot of insight into relatively simple systems that we can kind of set up that are variants of like oscillating networks interacting with each other. Question on the chat, thank you. So Shivank, how should we intuitively think about the order parameter? Does it measure the fraction of synchronized oscillators or something else? I think you can think of it as how close you are to having a synchronized network. I mean, in terms of the primary overall intuition. I've got a second question. So Shivank also says, okay, so different questions from Shirin. I'll get to your question as well. Yeah, it's just a tendency of the network to be synchronized. Okay. And what we'll discuss more in I think in the tutorial is like how that looks on the unit circle, and how that kind of just numerically looks as you play around with the values of K. But I don't think there's a kind of deep answer I can give, Shivank, to like how to intuitively think about the order parameter. It's really just a kind of description of the level of synchrony. But it's something that you can kind of measure, and it's how to say, I mean, it's a quantity that is often reported, right? So the actual value of K, right, depends on the model that you have. And that depends on the specific numbers that you're working with. But the order parameter in general is something that we can kind of compare across systems, and we kind of have the same quantity for different instantiations of Kuramoto model, and kind of have the same interpretation. So it's a generalization of something that we can give in the specific, language of some of the parameters of the model, but it will be specific to your specific model that you're using if you just look at those numbers. Shirin says, I remember when you mentioned the eigenvalues of Jacobian last time. Do we see oscillations? Does that mean we're dealing with complex numbers here? That's a good question. I'd love to see a graph on the complex plane. So Shirin, we've got some graphs on complex planes coming up. And then your question about Jacobian is also coming up. But yeah, the thing about Kuramoto is that it's not really bothering with that. This is my point about it being defined. I think the characteristic thing about Kuramoto is it's defined in terms of the phase. So we don't need to determine whether we have an oscillatory system with Kuramoto. It's defined as an oscillatory system. That is because it's, the derivative of the phase. It's like defined in terms of an oscillating, of a system of oscillators. So we don't do the kind of analysis that we will see coming up when we have other types of differential equation systems where we want to find if it's has an oscillatory, is it oscillatory fixed point, or what it's like at this linearization point. That's something that we do with other types of model, but not Kuramoto. Okay. So, okay. But how do you detect criticality if not on the complex plane? So the point isn't that we're not on the complex plane here. The point is that we're only on the complex plane. There isn't like the kind of activity variable starting point that we then use to find out whether we have oscillatory activity or not. We always have oscillatory activity with Kuramoto. Okay, hopefully that answers your question. Keep going if not. Shirin, thanks for the questions. Let's keep going. So that was network of coupled oscillators. Looking to data that we often see in recordings, and speaking to what I kind of kicked off when I was talking about Berger and oscillations and so on, is we see typically like a range of oscillatory frequencies, or power distributed across a range of oscillatory frequencies in most neural recordings. Again, at multiple spatial scales, they often look like this, right? So this is log frequency on the X, log power on the Y. And then the key thing here is it's a straight line. Okay, so straight line in log coordinates, log axes is what we call a power law scaling. Power law scaling in terms of the distribution of frequencies in the system here. So there's two kind of features here, I guess. One is again the just general trend of going diagonally down from the top left to the bottom right. Stronger power at lower frequencies, higher power at low frequencies, lower power at high frequencies. And then the second thing on top of that is little bumps. Okay, so little bumps that kind of deviate from the background trend of this 1/f. That will kind of indicate that we have some primary oscillation or some series of dominant oscillations. These kind of things are noise. If you have like really spiky blips in your spectrum, it's like almost certainly something like a line noise. But this is something that would be a kind of genuine say, strong neural oscillation, but it's embedded on this background here. So this background, this power law scaling in the frequency distribution, this is, we've got it showing it here for basically like one, if you like, data measurement on the left-hand side. And then this figure on the right-hand side is it's kind of an observation from Busacki, that the canonical ranges that more or less, like from a literature review of neuroscience, things that people look at in neuroscience, those also tend to fall on a log axis. Okay, so the kind of canonical frequency ranges that are reported and studied in the literature also kind of log like on a power law, show a power law kind of relationship. So there's got a comment at the bottom here. So we know, or it's kind of fairly well understood phenomenon in a variety of systems that this type of pattern where we have like 1 over f to some exponent. Okay, so the value of the exponent, this could change, and maybe it'll change from like 1 to 3 in the kind of data that you tend to see in neural systems. So that just determines like what the angle of the slope is. But it's 1 over f to some exponent. This is something that in other systems is understood as a kind of signature of critical systems in the vicinity of critical point, and potentially with hierarchy of interacting timescales or frequencies, or at least like multiple timescales and frequencies. So I wanted to kind of flesh this out a little bit, because this is something that you see this statement quite often. Don't see it unpacked too often. I'm going to try and unpack it. This is something I was adding in just before we kicked off, so see how it goes. So let's just talk about how we get 1 over f scaling and in critical systems. And yeah, this is like a conversation that I had with ChatGPT earlier today. But this is I think the key point here is basically having an autocorrelation function that follows a power law. So if we have some system that has a correlation function C, that like has a kind of critical value of its exponent. Okay, so it has some long decay rate. Then when we go in the Fourier domain on the the behavior of a like long autocorrelation function, and by long I mean temporally long. So if you have some time series that has correlations going out to long time, then in the Fourier domain, that's what gives us this distribution of this 1 over f distribution in the frequency pattern. Okay, so that just comes from the Fourier transform of the autocorrelation function, when the autocorrelation function has a kind of a critical exponent, or exponent that is going to kind of reappear over here as the exponent in our frequency distribution. Okay, so we'll come back. So the intuition is this idea of like long memory, right? As I mentioned, or broad timescales. So if you have activity in your features in your time series that kind of reappear, or this is what temporal, like long range temporal autocorrelation means, right? So you have something that is there's like basic kind of memory in your time series that extends over whatever, like a reasonably long distribution. Then that's the type of phenomenon that will kind of drive the 1 over f structure in the power spectrum. There's kind of more we could say about how that relates to the eigenvalues of the Jacobian as you move towards the critical points that we'll revisit at the end. But yeah, I just wanted to kind of comment on the 1/f part, and where that comes from, at least in terms of an intuition of what the properties of the time series are going to be. Yeah, so basically, if we have, say, no characteristic timescale or a range of characteristic timescales in our time domain activity variable, this is going to give us some scale-free dynamics in our power spectrum. Okay, now, so that's the 1/f. There's a lot more to say about 1/f. I'm not going to say too much more about it here, but we could talk more about it if people like. So I now want to just focus on some specific frequency ranges and oscillatory activity and things we know in certain canonical frequencies. I mean, in general, this is one of the things that is kind of principally of interest in clinical neuroscience. It's one of the features of brain activity that tends to be sensitive to pathologies and tends to be something that responds to interventions that we can do. And, you know, there's kind of scope for manipulating. So it's something that we want to be tracking. So for example, so this is synchrony. I want to talk about alpha yeah, okay. So this is an example from actually a little while back from Wolfsinger. So just looking at phase synchrony on scalp EEG in a patient group and a control group, and then looking at this over time. So these maps here, what you've got here is the average phase synchrony between about 60 locations on the scalp at each point in time, and for a range of frequencies over roughly 1 second following looking at a particular type of visual image, called a Mooney face, so a kind of black and white face. And the general idea, the general point here is like the kind of thing that we're looking at. I'm not going to comment too much on more of the details, just to say like there's a difference between these schizophrenia patients and controls in terms of the amount of synchrony that they're seeing at, it's about 200 to 300 milliseconds up to 400 milliseconds. So this is like the brain response to the face in terms of a kind of increase in the amount of synchronous activity between different parts of the brain at roughly 20 to 30 hertz. So when we're looking at these things here, these spectrograms, time frequency plots, you know, we've got something changing over time. We've got a range of frequencies on the Y, and we'll get some blobs. And the blobs are usually not super localized on frequency, right? So they'll span often something like a few hertz, often even maybe more than 10 to 20 hertz. In this case, it's like 20 to 30. So we call this low gamma or high beta. Okay. And then and it kicks in, as you see here, kicks in at 200 to 400 milliseconds following the onset of the stimulus. So less kind of beta gamma phase synchrony in schizophrenia patients than controls, and when they're looking at images of faces. This is like an example of the kind of things that we're looking at when we're thinking about like phase synchrony in neural data. In this case, like call it in Gestalt perception. So there's a kind of note of caution about looking at power spectra that we think about a lot in my group, which is to just not mistake a difference. If we kind of look in some specific frequency range and say we have condition A, blue, and condition B, red, and the specific frequency range, it's kind of normalized here, but this might be alpha or gamma, or like one of our canonical frequency ranges. And we see difference between two conditions or two patient groups or so on. If you don't look elsewhere, you might be misled to thinking that you have a frequency specific change, which I mean, it's kind of what we're looking at here. So if you think here, so these plots are kind of choosing a specific frequency. It's this beta gamma frequency, and then they're showing you the phase synchrony for that frequency, because we can't show multiple frequencies when we're demonstrating cross-channel synchrony with these kind of lines. We can only really show one frequency range. We can do multiple frequencies if we kind of collapse over space. So we have frequency over time here. So there's a range of frequencies here. So we're not kind of committing the error of kind of missing something that's a broadband effect, and thinking that that's something that's specific to a frequency range that we're looking at. But if we only did this here, then we could run into that problem, which is the problem we're making here. Like, you could have some broadband difference between conditions that if you only look in a certain frequency range, you might attribute to being some like oscillatory effect that is something that's not really properly understood that way. Okay, so we've got two examples to go through, two case studies that I want to focus on, that are the two kind of primary frequencies, rhythms that we really think about in neuroscience. Certainly two that I principally think about, alpha and gamma. Okay, so we're going to talk a little bit about alpha, talk a little bit about gamma. I already mentioned that alpha is kind of goes back to Berger, and is like one of the two things that Berger is kind of credited with discovering. So it's a pronounced large-scale 10 Hertz oscillation that you can see on the scalp. Power is inversely proportional to things like attention. Typical one is eyes open, eyes closed. So basically, you close your eyes for measuring EEG on your head, on your scalp. You're awake, and your eyes are open. Close your eyes, your alpha goes up. It's one of the most robust observations in all of neuroscience. This kind of states-dependent or kind of arousal dependence of the alpha amplitude. And that's what's being shown in this diagram here. So these are kind of blocked periods of eyes open, eyes closed over time. Okay, so you've got and eyes closed is the I, eyes open is the E. So, and then this is the time frequency plot like we had on the previous slide. So the main point here is, like, in these periods when the eyes are closed, we have this strong emergence, this strong frequency component around 10 Hertz. I mean, it's actually there with eyes open as well, but the frequency scale, the amplitude scaling here is just kind of emphasizing the change between the eyes open, eyes closed blocked. But yeah, this is something that kind of the most easy and, I guess, characteristic feature of alpha is it goes up when you close your eyes. And we see it principally on the back of the head. So there's a variety of ideas, theories about the origin of the alpha rhythm. I think we've covered this a little bit already, but we'll discuss it here and kind of moving forward. One is that we have, say, a thalamocortical circuit where we have some neural populations in the cortex, and then we have some neural populations in the thalamus, maybe the thalamic relay and the thalamic reticular nucleus, and we have this corticothalamic circuit, and this circuit is basically purring away at 10 Hertz. Another one that is kind of related to this idea of traveling waves is that we kind of say drop the thalamus and just think about the cortex. That would be your kind of points here. And then we say that there's a circuit within patches of cortex that will generate an alpha rhythm, usually with some well, yeah, always with some kind of inhibitory feedback loop. And then that might have some additional spatial structure over the brain, over the cortex that kind of will enable things like traveling waves as well, that we can also understand in terms of neural fields. A question, okay, thanks. So, question from Shirin, can you say something about what data sets you use in your research group and whether any of these are open source? Yeah, that's a great question. Shirin, if you can hold on until we start the tutorial, then we can talk a little bit about data sets. And yes, there's plenty that are open source, and there's plenty that you can kind of jump in and use without having to ask politely for people to share their data with you. I'll try and remember to get to that when we kick off the tutorial bit. Okay. So let's talk a bit more about alpha. So alpha, I talked about kind of the canonical resting alpha. We have eyes open, eyes closed. We also have this phenomenon called event-related changes in frequency. So event-related synchronization, event-related desynchronization. These are a bit of a combination of changes in probably the synchrony of neural populations and their amplitude, but we kind of group them together as just event-related synchronization and desynchronization. There's kind of a longer conversation about EEG, MEG methodology that I don't want to go into now. But the general point with ERD, ERS is that we can, like we saw in the previous slide with the frequency changes in response to the visual image, we have some change in the power in certain oscillatory frequencies that happens over time and can fluctuate over time in response to some stimulus. That's what ERD, or the converse of ERD is called ERS, so event-related synchronization. So it looks like, just to give you some examples, right? So this is actually a kind of flip color scale here. So red is going down. So visual tasks will suppress the level of alpha in the visual cortex because alpha is kind of an idling rhythm. So we can get event-related synchronization in visual parts of the brain during visual tasks, kind of similar thing with motor. And so we can think of them as being local, or like reasonably local, in the sense of these kind of scalp topographies. Okay, this is a motor, kind of left motor and right motor as well. So like in terms of like broad swathes of certain parts of the brain that we can kind of think about as certain parts of the head, we have localized changes in the distribution of power at certain frequency bands. Let me call that if ERD, ERS. So as I was saying, the general idea and kind of mechanism of alpha, or proposed idea about alpha, is it's an idling rhythm, okay? Which comes with this additional idea or like implication that what it's doing is it's kind of inhibiting. It's a kind of ongoing suppression that has a kind of converse relationship to active firing. So for example, this is also event-related synchronization and desynchronization plots on the scalp. And this is mapping it with a little bit more spatial detail in terms of what the hand area and the foot area for a sensory response, some to motor response. And so basically you see the kind of foot spot is kind of a little bit shifted over from the hand spot. That's just the kind of structure of the sensory homunculus over the motor cortex, but we have a kind of localized change in the power for kind of tickling the foot. It's like a little bit over from localized change in the power for tickling the hand. And this is what I mean by a kind of localized, or the way in which ERD and ERS power changes show some local modification of the amplitude of oscillations in certain brain areas. Yeah, so there's this idling versus active aspect to alpha. Idling is the high alpha, active, low alpha. They kind of flip-flop. And what kind of comes with that as well is this idea of gating. So this is like one of the concepts of how alpha can be kind of functioning from a cognitive point of view. Would be essentially like periods when we track the alpha phase over time. So here we've got a plot of a kind of fluctuating, this is like about 100 milliseconds, right? 0 to 100 milliseconds. The period alpha is a 10 hertz rhythm, remember? So the period of alpha is about 100 milliseconds peak to peak. Okay. So we have our kind of phase troughs and our phase peaks over the oscillation period. And one of the ideas around what the functional purpose of alpha is that in the phase troughs, you basically have less sensitivity to say external stimulation and sensory input and so on. And in the phase peaks, you have more sensitivity to incoming sensory information and so on. So you have a kind of modulation of kind of windows of attentional gating that can be kind of co-opted by cognitive processes to kind of structure the like packeting of information like from the environment into the brain. So that's the concept. And then the kind of thing, the way that we see that, and this comes back to the image I showed you with the schizophrenia versus controls and the phase coupling, although that was gamma, not alpha, but more or less same applies, is we can kind of track the phase coupling, and then the idea is that the phase couple will kind of see differential phase coupling along the periods, or the high and the low phases of the alpha phase cycle. So this is a general idea about how we kind of think about alpha in terms of giving us a mechanism for gating cognitive inputs. Another example of this, which I just kind of talk about briefly, is this kind of sound-induced double flash illusion type experiment, where basically you kind of give stimuli within, if you provide stimuli within a certain range of each other, a certain lag of each other, then it reduces their differentiability. So harder to kind of detect differences when they're kind of close, and that seems to relate to some extent to the temporal duration of the alpha phase envelope. That's the point that's being made here. Okay. So as I also mentioned, so those are some of the, say, cognitive studies and ideas around the nature of alpha. We also have things like changes with pathology, disease, illnesses, aging. So there's a few ideas that are worth commenting on here. One is about segregation and integration. Okay. So this is something that's actually, it's kind of common observation in neuroscience recordings. What kind of pointing to here is, so this is connectivity across a bunch of brain regions for different age ranges. And the point that would emphasizing here is this kind of band here, this rectangular band that's going, it's getting more bright. Oh, sorry, it's getting more red. So we get a kind of increased amount of correlated activity as you get older. And this is understood as kind of decreased segregation, overall increase in correlations of brain activity over kind of decades of age. Now, coming back to alpha, or coming back to frequency distribution as well. So this is from this study from Mazzini from a while back, just comparing three groups that are kind of age-controlled adults, people with mild cognitive impairment, people with mild Alzheimer's disease. And then the plot is power at these canonical frequency bands. So the point here is basically mainly here, okay, which is we have like lower levels of alpha power, also these lower frequency theta delta power, particularly the back of the head, lower levels of these in the patient group than the control group, right? So there's kind of less red here than here. So this type of observation that we see here, see in other types of data, quite a broad, like, common observation I was saying, is that reduced oscillatory power as a function of neurodegenerative disease. So there's power, there's also frequency. So alpha peak frequency, this decreases with age. This is a very robust and kind of widely commented on phenomenon. We've done some recent work on this as well. So it broadly looks like this. If you take the power spectrum, you kind of bin it over ages. So we have several deciles here, kind of 10 to 70 years old. You get a decrease in the alpha frequency. So the alpha is the value of the peak, right? The frequency value of the peak. You get a decrease of a certain percentage of a hertz every 10 years or so, every 10 years. And the endpoint being a difference of about 2-3 hertz from young adults to old adults. So basically your brain slows down as you get, literally your brain slows down as you get older. And this is related to things like working memory performance, but not in a way that's fully correlated with the age effect. So yeah, slowing down is something that seems to be a characteristic feature of oscillatory changes, particularly alpha rhythm changes in variety of neurodegenerative diseases. We talked a little bit about Alzheimer's, MCI. There's others we can talk about as well. It's a hallmark of globally disorganized or lesioned brain activity. This is one of the general ideas we have about the nature of oscillatory dynamics, and specifically kind of alpha rhythms in the brain in pathologies. I'm going to move on to gamma now. I think, yeah, so let's move on to gamma. So we talked about alpha. Alpha is, say, the most common frequency in the brain. Broadly speaking, gamma would be number 2. Depending on who you speak to, they might kind of flip them, right? Basically, if you're talking to someone who works with monkeys or maybe surgical data or mice, they might kind of put gamma first. And people like me will put alpha first, but you know, it depends on what you're working on. So the same thing, like with gamma, we want to see some peak in the frequency spectrum. Usually with gamma, there's kind of 2 types of gamma activity that we think about. There's, say, low, more kind of focal and like clearly rhythmic gamma activity, where we get a spike in the frequency spectrum around some specific value, like 40 hertz. And then the other would be what we call broadband gamma, where you have something that's more kind of a general shift over some large frequency range, like 40 to 80 hertz, for example. So the ideas around the, say, circuit origins of gamma are more kind of clearly in the area of these simple circuit motifs. And we have these acronyms that are used quite commonly in computational neuroscience, kind of denote these two ideas about the kind of circuit origin of gamma activity. So the first one, or the second one here, ping. So ping stands for pyramidal interneuron gamma. So gamma oscillations generated by an interaction between pyramidal cells and interneurons. That's what ping means. Now, pyramidal cells are excitatory, so they basically are E population here. And then interneurons is a bit of a vague concept, but often interneurons are inhibitory. So the interneuron in this case is inhibitory. So the idea with this concept of a ping mechanism is pretty much literally just what we think of as an oscillation generated by an EI population of the kind that is well described by a Wilson-Cowan model. So the kind of the feature, and there's been a good amount of experimental work kind of verifying that this is pretty correct, essentially, is that the oscillation is coming from this inhibitory feedback loop. The frequency of the oscillation, right? So the gamma frequency at 40 hertz is due to the time constant of the inhibitory synapses. That's the part of this kind of simple motif structure, but like there are neurons that represent this little sketch. The part of that circuit that is the slow bit, essentially, that gives you the delay in the feedback loop. The slowest part of it is the response of the inhibitory neurons. Sorry, the time constant of the response of the inhibitory neurons. So the kind of general thinking is that that's the clock cycle of gamma, or the clock cycle of gamma, like localized gamma, is kind of driven by the rate constant of your inhibition. Ing up here, I honestly don't know whether ping or ing came first or what the deal is with the history here, but it's kind of a similar idea, but without any E. So the idea is like you can have a network of interconnected neurons that are all inhibitory, and you can generate oscillations within a purely inhibitory network. And then the thing that matters is I mean, it's the same kind of thing again. It's the time constant of the inhibitory connections, but now it's the kind of inhibitory to inhibitory connections rather than the inhibitory to excitatory connections. And the reason why this is, I guess, kind of in the common consciousness is that there are definitely certain circuits that we can see, for example, in the hippocampus, that can be kind of identified to be ing structures rather than ping structures. They'll generate oscillations without any excitatory drive. But those are your kind of two primary ideas about gamma activity generation. A couple of questions in the chat, thanks. I'll mooch over there and pick them up. So some of the ideas about the kind of cognitive implications and cognitive functions of gamma are just skimming the questions on the chat here right now. So, okay, so Shirin says slowing down or slowly falling out of tune. I guess you were saying about how gamma slows with, so how alpha slows with age. Slowing down or slowing out of tune. I mean, it depends what you mean by in tune with that metaphor. You're definitely slowing down. Yeah, I think I see where you're going with that, but I think we would need to put a bit more meat on it. Sharon, for your comment there. Chris says, "Hi, Professor. Hope you're doing well. What is the main difference between alpha and gamma activity?" That's a good kind kind of broad question. Let me finish up on gamma, and then I'll try and kind of wrap it up with some comments on answering the question, what's the main difference between alpha and gamma? So with gamma, like the general space that people are thinking about, neuroscience is thinking about in terms of its contribution to things like neural coding, is around things like neural coding, but from the oscillatory point of view, it's things like temporal binding, temporal integration. So that but actually, yeah, I want to talk about this first. So this is something that we can see. This is more like an amplitude effect. So if we measure gamma activity in these, so this is in humans. You can tell it's in humans because it's human brain. Picture on the left. So this is from ECOG recordings in patients looking at visual stimuli, and these are visual stimuli that are different frequencies, like spatial frequencies, and the gamma response in different parts of the brain is kind of driven by the level of salience of the visual stimulus. So this is shown a little bit more clearly here. So these are four types of kind of stimulus SNR parameterizations. You see the image up at the top here. So this is saying, I'm showing you a grating, and I'm going to make the grating. So you're kind of looking at a screen, and there's this little circle in the middle that's got a few white and black lines, versus I'm looking at the screen, and then there's a larger circle with a few white and black lines, just increasing the size of the kind of stimulus from the center of the screen to a larger one. That's one. Then the second one is noise masking. So this is like adding in noise, so it becomes more speckled. Changing the contrast from a low contrast to a high contrast. And then the last one, which is a little bit different to the first three, is actually changing the orientation, but keeping the kind of SNR the same. So in each of these, what we're doing is what these guys are doing, G et al., they kind of parametrically varied this manipulation. So increase the size. These are different values of the size of the image. Same with the noise level, etc. Okay, so a couple of different points here on these plots. And then the thing that we're looking at is the amplitude of the amplitude of the gamma power, which is the kind of white line, and the frequency of the gamma, or the peak frequency of the gamma, which is the turquoise line. And then on the right here, we've got spiking responses as well. So the general point across all of these, more or less, is that each of these are examples of kind of amplifying the stimulus, but they're amplifying the stimulus on certain different axes of stimulus salience, let's say, right? Like size, contrast, noisiness, orientation. So in each of these cases, the kind of gamma amplitude changes parametrically with the effect, right? And basically goes up. So if you increase the size, the gamma amplitude goes up. Increase the noise level, gamma amplitude goes down. So decreasing the salience, right? Increase the contrast, increase the salience, amplitude goes up. The last one is slightly different, so we'll talk about that separately. And this tracks, in most cases, but not all cases, the spiking activity. So this thing on the right here, this is like the amount of neural firing. Okay, so unlike, one of the take-home points here is that unlike alpha, and this will be one of my answers to the question you asked, Krish, as well, which has this, broadly speaking, kind of anti-phase relationship with things like cognitive load, or this kind of push and pull relationship with cognitive load and like engagement of cognitive tasks. It's an idling rhythm. So if you stop idling, then your alpha goes down. With gamma, it's the opposite. Gamma, broadly speaking, like when you increase the salience of the stimulus, or you increase the kind of the activity level, for example, the spiking rate of the neurons in that patch of tissue, the gamma goes up with that. So gamma is kind of associated in a kind of tight way with the level of neural firing. And yeah, the frequency band plots, the power spectral plots on the left hand side, just kind of showing what we see over there. So for example, like the power going down or with the increasing noise level, and going up with the increasing stimulus salience and so on. So gamma has this linear relationship with, or proportional relationship with, neural activity and neural firing. Which we can see again here. So firing rate, frequency, firing rate. I want to see, yeah, we'll skip that one. So this is the same point again. So spatial and temporal coherence. I think we can kind of move along with that one. So that I showed you a bunch of things about gamma power. We can, like we saw with the very first example with schizophrenia, we're going to have everything. We're going to ask about changes in power, like localized changes in the amplitude of some oscillation. You know, when we're kind of seeing those locations in the visual system with those visual responses, or in the case of the alpha with the somatosensory response going down, alpha going down for when you kind of tap the finger, or so on and so on. So we think about localized power. We can also think about coherence or synchrony between different brain areas. And one of the ideas that has had a lot of currency that connects to that is this idea of communication through coherence, right? So the idea here is that you have some kind of baseline oscillation that is common between two nodes in a network. And if they're synchronized, and also synchronized with zero lag, or synchronized with zero lag, probably the best way of describing it. Then, so this is, I say, some base frequency, and then what that allows then is for activity here kind of influence activity here, and kind of bidirectionally. So these guys can kind of talk to each other if they're synchronized, because the background oscillation is giving this, as we described before, this kind of gating operation where if you're on the dip, then you're kind of basically being suppressed. The window for communication is on the peak of the oscillation phase. So this is the idea of communication through coherence. Like if you can synchronize your oscillations, then when such that they kind of know about each other in terms of these communication windows, that's when you want to fire, and that's when you have a kind of line of communication. And that's a kind of understanding about the functional implication of synchronous oscillations in terms of a role in neural coding. So, I mean, this is how that kind of pan out in terms of a network structure. You know, so we have some sensory input that is being kind of converted into some firing rate, okay, and we have some other sensory input that's being converted into some firing rate. So we have these different sensory inputs that are kind of converging on some higher level pathway, and the general idea is like if these guys are, if there's some clock cycle, which is basically what the idea of this kind of carrier wave is. So have some clock cycle, and that gives us a language, a time a kind of time structure with which we can integrate different types of information and information coming from different sources. Okay, so these are some of the general ideas around, summing up, like oscillatory coding, particularly in the case of gamma. It talks about things along the lines of communication through coherence. This is also called related to it's more of a kind of cognitive concept of binding, combining different parts of different pieces of information. Okay, so you have information that, say, the visual and the auditory parts of a complex stimulus. Ultimately, you want to combine those into some common percept. So you'd need to combine information from different parts of the different sensory systems. So that'd be an example of when you need to do binding. At a more granular level, we talk about kind of combining things in terms of time and in terms of phase. That's what this idea is here, right? So combining things on the phase of some carrier wave as a general mechanism and role of coding from the point of view of oscillations. And the units, the different nodes of the network are kind of distributed potentially quite widely around the brain. So we think of these in terms of distributed cell assemblies. Okay, so this is just another sketch of like different variants of the ping-ing motifs that I talked about before. The final, I think, 2 slides before we wrap up, and then I'll get to the questions that you had on the chat, and then we can have a quick break. Is just revisit some of the analysis that we did a few lectures back, which was one of the questions we had earlier on about the role of the Jacobian in terms of determining whether or not we have some oscillatory activity or not. So processes by which oscillatory solutions emerge in different models that we can study and systems that we can study. They commonly, they almost invariably involve interactions between excitation and inhibition, or maybe time delays, and some combination of those things, potentially with different time scales. The things that we can, so we can think about different, say, mechanisms, neural mechanisms leading to certain types of the structures that we need to give us oscillatory activity. We can have inhibitory feedback. Okay, so this is one of the things we talked about before. I talked about all the way through, like the eye population feeding back, giving us those ping oscillations, or the thalamic nuclei feeding back on the cortex, giving us a kind of slower inhibitory feedback for alpha. Adaptation is something that can lead to some kind of oscillatory structure, in the sense of you have some time varying change in the response that can lead to some kind of temporal signatures that can be oscillatory, and relaxation oscillations. So when we have some system that has a fast and a slow component. We thought about this, I think, when we were talking about in the very first lecture, when we were talking about Fitz and Nagumo and Maurice Lacar and Hodgkin Huxley, indeed, right? So Hodgkin Huxley is a good example, right? So we have oscillations in Hodgkin Huxley equations, mainly coming from the fact that there's this potassium component, right? This potassium conductances that are giving it. They're our slow recovery variable in Hodgkin Huxley, and in our simplifications of Hodgkin Huxley, and they're the kind of part of the system that that combined with the fast excitation in the case of Hodgkin Huxley, mainly driven by sodium, the interaction of those two is something that can lead us into having some oscillatory activity at the single neuron level. So these are structures that can lead to oscillatory activity. Now we looked at Wilson-Cowan, quite a lot at different points, and we'll keep going with Wilson-Cowan today and in later lectures. And I think we looked at we certainly talked about the concept of linearizing around a fixed point, and then thinking about the eigenvalues of the Jacobian for say, well, in general, but for several like two-dimensional dynamical system models for neural systems. So with Wilson-Cowan, this is kind of how it pans out. So we have our Wilson-Cowan equations, just to remind folks. So we have our time evolution of excitatory population, time evolution of inhibitory population, driven by self-feedback, and then the input from a self-connection and the input from a connection from the other population. And in the case of the input from the inhibitory population, it's going to be inhibitory, so we've got a negative here. And for the input for the excitatory population, here and here, it's going to be positive. And then some background, some external drive as well. So there's plus I. So Wilson-Cowan equations. So we can find some fixed points, or choose or identify some place that we want to characterize the dynamics around, that we want to linearize the system around. So we can define the Jacobian for Wilson-Cowan equations quite simply in terms of the model parameters, right? The coupling weights and the time constants gives us this two-dimensional matrix. And then the stability of the system at this fixed point is determined by the eigenvalues of this matrix. So we can, so we're going to look at this in the tutorial. Paz will pick it up. The simple thing to do is to just do eig on that matrix when you plug in the numbers. But we can identify the eigenvalues analytically or numerically. What we care about is whether they are, well, if they are complex, and when they are complex, what the values of their real and their imaginary components are, right? So the real part basically gives us the amplitude, the imaginary part gives us the frequency, and then oscillatory solutions will emerge when the eigenvalues of the Jacobian are going to be positive. Okay, so the imaginary part. So this is something that gives us some, well, we're kind of following this through. So we talked about the scaling of the amplitude and the frequency in terms of the power law scaling. Okay, that's something that we won't be looking at, I don't think, in the tutorial, but we can kind of maybe pick up in later lectures, like how the distribution of the eigenvalues can control the shape of the power spectrum in terms of those frequency scaling, those power law scaling properties that we were talking about earlier. But the general message, just coming back to what we had, I think, excuse me, over here, and this will lead us into our tutorial as well. So the in the Wilson-Cowan equation example, which is kind of our workhorse, but also one of our simple examples that we can study in a lot of detail, we can break it down, choose some fixed points, and evaluate the stability, and then look at understand the oscillatory properties of a EI network with some analytic and numerical computations. We're going to look at those in the tutorial coming up, and then coming back to what we had over the last few over the last hour. Just to summarize, so touched on Kuromoto model. Again, Kuromoto model is a a workhorse in computational neuroscience, and it's kind of the simplest possible model of interacting oscillatory networks, and more broadly, more or less the simplest possible model of oscillatory brain structures, full stop. And there's a lot we can do with Kuramoto, but generally speaking, we're interested in how the network structure determines the synchrony level across a network. And then just think we talked a lot mostly about data and the kind of nature of oscillatory activity in brain measurements. Alpha and gamma being the principal frequencies that are of interest, broadly speaking, in neuroscience. The difference coming to, I think the question, Krish's earlier question, what's the main difference between alpha and gamma activity? So I'll answer that now. I mean, the main difference is they're different frequencies, okay? But I assume that's not a satisfactory answer because that's kind of obvious. But I think I mentioned right at the start that we tend to look at gamma as more locally than alpha, by which I mean, well, one of the ways that that's the case is that the data that where you see the most gamma and the most good gamma tends to be when you have some experimental setup where we are kind of recording things that are like sub-centimeter. You've got a kind of small electrode that can pick up a local field potential that doesn't have too much kind of spatial width. So again, those tend to be invasive recordings in human patients or in non-human primates or things like that. So gamma tends to be more local than alpha, or at least the most interesting things about gamma we can see in a more spatially localized domains than the things we see with alpha. Alpha tends to be, we see stuff in different parts of the brain. We see stuff at the back of the front, and we can kind of look in the motor cortex, and we can see certain things that are like somewhat localized. But even the things that we can see that are somewhat localized, they still cover quite a lot of the general area. So yeah, alpha is more global, gamma is more local, and then the different frequencies. And then I think the other kind of key difference between alpha and gamma, as I was saying earlier, is that broadly speaking, neural activity goes up, alpha goes down, and with gamma, it's the opposite. Broadly speaking, neural activity, specifically firing rate, the amount of neural firing, and relatedly, the amount of metabolic activity. So if you kind of translate this over to fMRI, where you're looking at the kind of metabolic signature of neural activity, that's usually going to track gamma more than it's going to track alpha, because gamma tracks neural firing, not inversely, but will go in the same direction as neural firing. And that's one of the, I think, biggest differences between alpha and gamma, broadly speaking, in terms of how it behaves in measurements that we make. Okay, so that's it for our intro lecture, and questions from the group, after which we'll take a break, and then Parza will pick it up with the tutorial on Colab as usual.
So I'm really analyzing kind of how are there ways that we could look at neural activity and as we're getting our feedback, say like the brain is currently in a gamma state or alpha state, where the probability that we're observing one of these frequencies is quite high, because we also briefly went over when it can like stop the frequency from happening. I don't know if we touched upon that too much, but I was wondering, can we do the three things, say, what is the probability, kind of, or like a probability measure, or just, if we're going to approach an alpha state, if we're going to change states, we'll go to a gamma state, and then that change would be I know that's not the
Yeah.
only three states, but I'm saying like.
Yeah, no, it's definitely the right line of thinking, and I think I can kind of say two things about this. One of them I don't have slides for, and it's new, which is cool because you're asking the right kind of question. But I think this is where you're getting at, right? So this is kind of moving in and out of high alpha, a high alpha state. So I think the first point I want to make about this, just talking about, say, changing alpha over time. So this would be like the simple experimental context for this is like eyes open, eyes closed, as I was saying. So it depends on a few things. One of the things, it depends on just intersubject variability, how much you like literally kill the alpha rhythm when you open your eyes. So the general phenomenon, if I've got the power spectrum around here, eyes open, eyes closed. Oh, do I have eyes open, eyes closed, power spectrum? Oh, I don't have this. I don't have the image I want. But if you think about sorry for flipping around. Yeah, here. Okay. So if you think about the power spectrum, kind of that will correspond to these two, it's both going to be this like 1/f. Yeah, I'll bring up an alpha power spectrum just to kind of be clear. So you have this. This would be, and this is like a very strong alpha.
It was earlier in the slides. Slides the one little bump in the noise on the bottom.
Yeah, we could use that one as well. Yeah, we could use that, but I don't think I think that was on like log x-axis. This will do. So this would be like high alpha, eyes closed. Okay, and then eyes open would be basically look the same, but the peak would just go down, right? So that's what we mean by the eyes open, eyes closed, and it's called alpha blocking. And it's also event-related desynchronization from that kind of lingo. Now, the extent to which, say, you versus me versus Jeff and Susan has like the magnitude of their alpha blocking for, and you can do alpha blocking for things other than eyes open, eyes closed. You get alpha blocking in the motor cortex, which is called the mu rhythm, which is also an alpha frequency rhythm. If you if you tap your finger, okay. So the resting motor cortex is also resting at alpha, and then when you engage the motor cortex in the same way that you engage the visual cortex when you open your eyes, you engage the motor cortex by tapping your finger. You also suppress the roughly 10 hertz rhythm in the motor cortex when you have the active, the active task. So the alpha blocking is something that kind of happens wherever there's alpha. The extent of alpha blocking depends on the individual. I mean, it depends on like the magnitude of the change to some extent, but basically, like some people have complete alpha blocking where you compare eyes open, eyes closed, and there's just like the eyes open is like almost flat, and then other people, you still got a bump. So what that means, I mean, it's kind of hard to say really, like the significance of complete versus full, but you're asking about moving between states, right? So part of the answer to your question there is like the extent to which you move between at least the high and low states is something that, just in terms of the things that we definitely know, which is like the data we can measure, there seems to be some level of variability. Now, the other thing you're saying is moving between, say, alpha state and gamma state, and that is also part of the picture here that I haven't really emphasized, but I mean, it's kind of implicit, at least if you take kind of case study 1, case study 2. So alpha and gamma are kind of anti-correlated as well. The tricky thing about this is that they can be anti-correlated in 2 ways. They can be anti-correlated in if you kind of take this out to the gamma and you imagine this is a higher gamma, you imagine this is something that's maybe ECOG rather than a scalp EEG, where you can see a good clear alpha bump, sorry, gamma bump. then what you'd be interested in is like whether your alpha bump and your gamma bump are anti-correlated, that, you know, this goes down, this goes up, right? And broadly speaking, that's the type of relationship that you get between alpha and gamma, because gamma is like high activity, alpha is idling. But there's another way in which they're anti-correlated, which is a bit of a comes to the point I was making about not being misled by localized band power changes, is that you've also got the background slope. So changing the background slope, if it kind of seesaws, then your low frequencies go down and your high frequencies go up. So there's two ways that you can kind of achieve anti-correlation between low and high frequencies. One is from a kind of, feel like, legitimate suppression of a peak and a legitimate enhancement of a peak. And the other is just a change in the overall shape of the 1/f. And so definitely alpha and gamma are anti-correlated, but whether it's which one of those two processes is the right way to understand it is, I think, is still an open question. Final comment on what you're saying about like transitioning between states. So this is, maybe I should just send a reference around. So there's some recent work from a group in Oxford that is led by a guy called Mark Woolrich that has been looking at exactly this. So it's basically tracking, there's this thing called a hidden Markov model. So it's kind of a somewhat fancy signal processing analysis that does some kind of ICAs on a bunch of different channels, but the endpoint is they're looking over time, over usually like hundreds of milliseconds, and then looking for changes in brain states they can define as like distinguishable clusters in a kind of more spatial time frequency cluster components, and they have temporal signatures. So there is a cycling between alpha dominant state and I think a beta gamma dominant state. I forget like exactly what has been come out of these results, so I can pull up the paper for you. But the interesting thing is that, yeah, there is, if you track resting state and you look in a kind of global way, like global across space, as in across brain areas and across frequencies, then there is like switching between states where some frequencies are dominant at some times and some frequencies are dominant at other times. And it's on a slower kind of time scale than the other fluctuations that I was talking about with the coding idea, right? So the coding idea is kind of faster. This is like, I've got some information, some sensory information. I've got some slow frequency carrier wave. This could be alpha. And then I'm gonna fire fast and kind of bind information from these two locations, from these two brain areas, if I can make sure that they align on the peaks of their phases. Okay. That's much faster than the slow variation in the dominant frequency states that's coming out of this like newer work from the OHBA group in Oxford, but I can pull up some references for that if you're interested. But yeah, you get kind of changes in the dominant frequency. But broadly speaking, in human EEG, like it's always alpha that's dominant, but there's like the composition over the entire spectrum does fluctuate. If you just Google like hidden Markov model MEG human brain, then you'll probably get a lot of stuff on that. It was introduced about 10 years ago from this group in Oxford by a guy named Mark Woolrich, Woolrich. And it's one of the more, I think at this point, one of the more commonly used tools for like resting state analysis of EEG and MEG data. And yeah, it's kind of based around the idea of trying to find what are the primary states and what is the transition between them, and then also thinking about transition probabilities and dwell times and things like that.
I have a lot of background in financial mathematics, and that's where it pulls from these time series with hidden Markov models. It's we want to know when does the time series look like it's idling, like the stock price might not be moving so much, and when do we see a big boost in liquidity, or what is the energy, the big gas nights coming in.
Okay.
And so that's why I'm asking this question.
Oh, yeah.
I'm curious because I know hidden Markov models can be used to try and find the states, like you're saying, when does this asset price or this time series start to change a lot? And then can you tell me the probability of that? Because then I can profit off that.
Yeah. So right. So I don't think I can tell you how to profit off any of this. Yeah. I'm not sure I'd tell you if I knew. But you have a transition matrix. So you have a transition probability matrix from this kind of hidden Markov model approach, and it tells you a little bit more than just knowing that there's clusters. It tells you like, okay, well, when you're in this state, then you're going to go to this state next, and there's a sequence. And that, the addition to that of like the cycling and then the identification of the frequency components and the order in which they cycle around is something I think was like maybe 2 or 3 years that there's been a lot of work on that, so it's quite fresh.
I wonder because it seems very similar where you have like kind of these stochastic processes, these time series where it's like neural activity, right? And you're trying to find some sort of pattern or like what we're saying, oscillatory behavior. And then yeah, I don't know. I find it very interesting, like the crossover. I think there's not very much, but there's tension there that I feel like should bridge the gap between like financial mathematics, neuroscience, that way, because the choices of the economy come down to the brain activity of individuals in a stochastic way.
Right.
And so,
well, that's really bridging the gaps. That is.
yeah.
Go
So I wonder
from brains to markets.
Yeah, I wonder how much works with that.
Yeah, but I think I mean there are like I think there's been a decent amount of cross-pollination. No, I don't know if cross-pollination, but input to kind of signal processing approaches in particular in neuroscience from like time series analysis in general, and time series analysis in general often comes from econometrics, right? Like Granger analysis is and MVAR modeling is the obvious one, and even complex systems, right? And looking at fluctuations in time series as from a kind of nonlinear dynamics point of view, like that's been a feature in neuroscience and been a feature in, I guess, certain parts of mathematical finance for a while as well. So I think there's often things that kind of crop up in, and same geology, right? Or and ecology. Like there's a few fields where certain things like, oscillatory properties in time series, networks, fluctuations, dynamics, nonlinear properties, they kind of have applications, and neuroscience is usually one of them as well. Cool. Okay, so we've got, I think, one last question on the call, and then we'll call it and then we'll take a break. So last question from Shirin. Do different values of the eigenvalue, say real part positive, negative, or 0, point to any critical lines where we get decoherence or phase transitions, or are the eigenvalues used mainly to pinpoint different oscillatory behavior? So what would be kind of neuroscience analog of decoherence? I see where you're coming from, Shirin, and I think you've raised some ideas using some concepts from physics and kind of seeing what the analogies are. I think decoherence, it's not obvious to me what what to
Oh,
point
well,
to
just
from the.
desynchronize, like when you have oscillations. I mean, I'm just looking to see if the eigenvalues are like positive, the real the real part is positive, but like how does that affect the oscillations, and is there any difference when there is zero or negative? And then do we see like phase transitions, or if you like desynchronization between um, the system if the values are either positive, negative, or like zero. Sorry, I said it in a very complicated way, but I'm just trying to see like what causes this desynchronization,
Yeah, yeah,
or like the slowing down. Like when does it happen? Is there like a critical line in a sense? I mean, that's a bit of number theory analogy,
right.
I think, but yeah, sorry about the complicated question.
Yeah, so I mean I understand. I think it's a very kind of similar to the question you're asking before, and so I think the answer is kind of similar, which is yeah, maybe the best example of the thing closest to decoherence and the opposite kind of emergence of synchrony from the stuff we've been talking about here is the Kuramoto model. I don't think Kuramoto model can give us loss of synchrony. I think you evolve towards synchrony and then you stay there. But if that works for you, like the opposite of decoherence, like the evolution of coherence, then Kuramoto model would be the way that that's thought about from most of the computational neuroscience point of view. But again, that's not really to do with the eigenvalues of the Jacobian in the way that we were looking at it with the Wilson-Cowan equation. The Wilson-Cowan equation, linearization there isn't really telling us about synchrony. It's just telling us about the amplitude of oscillations, the presence and the amplitude and the frequency of oscillations, but not about synchrony in the sense of multiple units. Now, yeah, there's other stuff we could do that I think goes in that direction, but in terms of what we covered today, the Jacobian stuff is not about synchrony. The Jacobian stuff is about oscillations. The Kuramoto stuff is about synchrony. I think that's my best.
Great, thanks. That was very helpful. Thank you.
Yeah. Okay. Great. Good questions as usual. So I think we'll leave it there. So let's take a break. We'll take a 10-minute break. The time now is 10 to 4. So we'll see you back here at 4 for a excellent collab-based tutorial that Parsa will lead, and we'll be delving. Oh my God, I'm talking like an LLM. We'll be diving into some of the stuff that we've talked about in the lecture in a little bit more detail. So see you in 10 minutes.
So 4 o'clock, let's get started with this tutorial. And as you guys are aware, today's lecture was about brain rhythms and synchrony and oscillations. And the tutorial for this section spans 4 different parts. The first part is just going to be an introduction, looking at what we mean by different ranges of oscillations. Second part is going to be on the Kuramoto model, which we briefly discussed during the lecture. We're gonna really sink our teeth into it to understand it better and understand what it can do for us. The third part of it would be something that you've already done, would be related to stability analysis surrounding a fixed point, like a dynamical systems analysis. And the last part is going to be oscillations at the level of neural patches, using neural mass models that we discussed. I think it was last lecture or the lecture before that. So I'm going to quickly go over the introduction because Dr. Griffiths has already spent a good time discussing each of the oscillation frequencies. essentially the brain signal, you can somewhat arbitrarily categorize into different frequency bands. You know, starting from delta, the lowest, as you increase the frequency, go from delta to theta to alpha to beta, gamma. And when you look at neural signals, you see something like this, a combination of all different frequencies, but with a preferential engagement or involvement of alpha frequencies more than the other frequency bands. So we'll get to alpha in later parts of the lecture, but it's got a bunch of interesting dynamics on its own. I'm not gonna belabor the points too much, so I'm just gonna go towards more into the interesting parts of the tutorial, starting with the Kuramoto model. So the Kuramoto model, as Dr. Griffith mentioned, it's a very simple and elegant mathematical framework to model and study synchrony and emergent oscillations. It's pretty straightforward, as you can see from the equations. And it's essentially telling you, it's describing the oscillations of different oscillators, different rings, with respect to the change of their phases. So what's happening here, and what this equation is saying here, is that the change in the phase of a single oscillator within this coupled oscillator system depends on its angular frequency, which is, that makes sense. The change of phase depends on its frequency of oscillation. That's pretty intuitive. So omega I is the natural frequency of the oscillator, plus interactions from the other oscillators are coupled to it. So what that means is there's a coupling parameter K, which is a coupling strength between different oscillators, and an average term, which is the sign of the difference between the phases. So essentially, this means that the oscillator is gonna, rotate depending on its own intrinsic frequency, and there's going to be some additional input based on how divergent it is from the other frequencies of other oscillators within this system. So let me actually show you what this looks like. Well, this is a static image, but let me give you the more dynamic image. So this is what I mean. So each color over here is one oscillator. And as you can see, they're oscillating at different frequencies. So for example, this green one here is oscillating pretty slowly. The purple one's doing it pretty fast. And what we've done here actually is that we've coupled them together. So they, depending on the parameter K, they're able to influence each other. So here in this example, we're saying that the coupling parameter is actually pretty low. So they don't really couple together. They don't really influence their frequencies don't really shift the frequencies of other oscillators. They're doing every oscillator is doing its own thing, sort of, right? But as we increase this coupling parameter, what we're gonna see is that, first they kind of start off doing their own thing, but after a while, they start to sync up. They start to behave as a collective, and a collective oscillation will emerge over here. Now, if we increase it even further, what we'll see is two things happening. There's two things happening. One is you can see that this coupling, this synchronization, this emergent pattern of synchronization just pops up faster. You fall into that regime much faster, much more rapidly, and also that you're kind of like more cohesive. They're influencing each other more cohesively. And one way that we can actually also we can obviously see this sort of emergent oscillations happening. Yeah.
Is the radius now?
I'm sorry.
Is the radius now complicated?
Over here. Yes, exactly. So this was a question in the during the lecture as well, that how do we actually measure this coupling? So how do we define this coupling strength? That's actually the order parameter, it's R over here. And intuitively, what we can think of it is essentially like, how well do they align? Yeah. So it's just a sum of all the different vectors. And if they're aligned, if this black thing, the magnitude of this black thing over here would be the order parameter. The radius of the entire thing is 1, so it's a unit circle. But yeah, essentially what we're seeing here, for example, is that when each individual oscillator is pointing in its own direction, the sum of it is going to sum to 0. So what you'll see is like a order parameter that's somewhat near 0. But as they become more and more aligned, then this goes closer, and it gets closer and closer to 1. So when they're very much clustered together over here, then you have an order parameter to close to 1. So you can actually look at this, and let me just show you the equations for the order parameter. It's pretty straightforward. It's just saying that it's the sum of the phases. And yeah, so this thing over here with the order parameter, which you can calculate by essentially summing up the different vectors, the phases of the each individual oscillators. So what we're seeing over here is essentially the evolution of the synchronization of the order parameter over time. So over here, it kind of like something interesting is happening here. Over here, no matter how long we let this simulation play out, it will never get to a synchronization state. It just wobbles up and down, but it never really couples. They don't really couple together. And that emergent synchronization never really happens. But as we increase it past a certain value, certain threshold, we see that this kind of like, maybe right about here, the system sort of falls into this other mode of other regime, this sort of synchronization regime. And then it sort of stays here. Yep. It just wobbles around here. I'm not sure. Maybe, but I'm not sure, honestly. I wouldn't know. And you can see that, for example, over here, at this point, it's just fallen into this other regime. And for the high coupling case, this other regime seems to be like present from the beginning. It just sort of falls into this coupled states from the get-go. Now we can see this happening. We can see that intuitively when we look at it, we can see, okay, this coupling, this emergent synchronization is happening depending on, well, the coupling parameter, how strongly we're actually coupling them together, but also it also depends on how divergent or how heterogeneous are the distribution of the frequencies of the entire set of oscillators that are involved. If they're pretty much the same, they're oscillating around the same frequency, then it doesn't really take much to get them to synchronize. But if one is oscillating at one hertz, the other is oscillating at 100 hertz, you can just intuitively imagine that would be much harder to actually get them to oscillate at the same frequency. So you can actually look at that mathematically. We can look to see, how would this critical coupling, this phase transition, what would it depend on? So the formula for it is given here. I haven't gone into the derivation of it, but essentially what this is saying is that the critical coupling depends on the distribution of the underlying frequencies. Yep.
The force pushing together is just the sine of the difference. So it shouldn't differentiate between like 0 degrees and 180 degrees, right?
Sine. Yeah. So yes.
Similar thing. So why do we not see like?
Well, yeah, that's a good question. That you actually do see that. You actually might see different clusters of oscillation happening. Yeah, it's a good question. So it's because like it's only like 3 different examples, and you can actually, there's a widget, there's an exercise for it that you can actually like play around with different cases and see if that actually happens or not. So it actually, just a minor point on what you're saying, there's 2 different quote unquote forces that make this oscillation possible. One is a convergent force, which is given to us by this coupling parameter, and one is a divergent force. So the divergence force would be the difference, and the convergent force would be that scalar k that's kind of pulling them together. But like you're saying, like, if they're absolutely antiphase, they start up from antiphase position. And let's say like a bimodal distribution of, like, we have a bunch of different oscillators. Some of them are, just, peak is over here. The other ones are peak is over here. Then you might actually see a synchronized state with two different kind of like activity. Yeah, good question. So back to this type of phase transition. Let's explore this phase transition more rigorously, see like what it depends on. It's the frequency of the formula for it, the Kc, the critical K value, is 2 over pi g of omega, and g of omega is actually the distribution of the intrinsic frequencies of the oscillators involved. And so when it's a negative, when K is less than this coupling, less than the critical coupling, then you won't get that synchronization. It's similar to the first case that we were saying. Like, no matter how long you let it run, you won't get emergent oscillations. And when it's greater, equal to greater, then it actually starts falling into that other regime. So at this Kc value, it bifurcates into the synchronized regime. And let's see. And this synchronization, again, depends on the distribution of the underlying oscillators involved. So for example, here we have a Gaussian distribution and we have a uniform distribution. So the Gaussian distribution is, when most of the there's a center, there's an average, quote unquote, of the oscillators involved. And when that happens, you can intuitively see that they're already somewhat aligned. So it would take less for them to actually fall into that synchronized state. So the coupling strength would need to be less, doesn't need to be as intense. But if you have, like, for example, uniform distribution where it's just absolutely spread out, then you would need a more like convergent force to actually bring them all together. So that's why you can see that, in a uniform distribution, the coupling strength is going to be bigger. So this is an exercise for you to actually play around with the different parameters of K and the different distributions that you can get. And yes, so it's an interactive widget that you can play around with. That's a very brief overview of the Kuramoto model. Any questions so far? Any in the chat? Hi, have you uploaded last week's slides and collab? No. Okay.
Saturday, I'm doing that right now.
Yeah.
So,
Go.
yes, I think you'll see it there now.
Yep.
You go back, I think it's omega actually was the term used for the distribution, I believe.
Yes, G of omega. Yeah.
So that something that you get from the data, is that assumed? Is that what?
So you essentially set this up. So this is omega. G of omega is a distribution of the oscillators that you want to put in. So for example, as just a random example, let's say I'm saying my brain is made up of like 10 different oscillators, 10 different regions. Each of them is doing like something something related to their own activity. So in that case, I have 10 different oscillators, so 10 different intrinsic frequencies. So then the distribution would be a distribution of 10. Now, it might be the case that they're pretty much centered around the same value. They're all oscillating at 10 hertz. So you get a kind of like Gaussian type of distribution centered around 10. Or you might see like an absolutely uncorrelated activity pattern, in which case you would see a uniform distribution all over the place.
Could you sample this from like real brain? Like activity if you wanted to?
You could do that, and when you actually do that, it gets a bit more complicated, or not complicated, but a bit more nuanced, because what I'm showing you over here is a very homogeneous type of connectivity. So what I'm assuming here, I didn't explicitly mention this, but it's good that you brought it up. What I'm assuming here with this equation is that every oscillator is coupled to every other oscillator with the same connectivity strength, K, right? But that's not the case necessarily in the brain, right? Some regions are connected to each other more strongly, some regions more sparsely, right? So you can actually have an adjacency matrix for this. So you can swap out this K value for an actual matrix. So this, in that case, like, the adjacency would go within the integral, and you can actually model network behavior rather than just like a homogeneous type of network. Does that answer your question?
It does, yeah, and then I guess the other assumption is, the choice of frequency ranges as well, right?
Right.
You can arguably, if you wanted to change those,
Yeah.
like the ranges that way.
Yeah. I mean, you have control over that depending on like what your assumption is, what your underlying assumption is.
Right. And it's okay, and then so those frequencies specifically, those ranges, are those just kind of like the most popular or most like frequently observed kind of frequencies? So we have them.
Right here, honestly, what I'm explaining to you is not really even necessarily related to the brain. So I'm just initializing a random sample, a random distribution, a Gaussian distribution. But for example, you can apply this to brain activity, saying that in that case, you would, for example, say that, there's an alpha activity permeating in every region, and there might be a little jitter between different regions. So in that case, I would initialize my distribution around 10 hertz, but with some like, variance. Or you could say like, oh, I'm not actually modeling brain data. I'm modeling like financial dynamics. So there's like, I don't know, purchasing cycles, there's like, I don't know, marketing cycles, all that, right? And you can say, oh, maybe like, every 2 days people go out to buy whatever, but every like 2 months there's a marketing campaign or whatever. So it really depends on like what you're trying to actually model and what you're trying to do. You would have control over like, what you set your model to be. Cool. Any other questions for this section? Awesome. All right. Okay. So next section is something that you've actually been exposed to before, and hopefully, you know very well, because it was one of the, I guess, more challenging exercises during for the course. This is, again, mathematical analysis of the behavior of a system surrounding a stable point. So that was a Kuramoto model. We're just, let's package it up and we're gonna set it aside. We're gonna go back to the Wilson-Count equations. So the Wilson-Count equations, if you remember, there was population of excitatory and population of inhibitory, and we're characterizing them by the ratio of activity, the change in the activity ratios between different populations, within different populations. And if you remember, well, one way that we can actually study the behavior is, through simulations and through numerical simulations. Just we have the equations and just let it play out with a lot of computation, and then we just see what the behavior of the system is. But another way to do it is actually, to see if we can get some analytical solution just by the parameters of the model itself. So we're revisiting that really quickly, and Dr. Griffiths also mentioned that during the final few slides of the lecture. What we're actually doing here is we're looking at the behavior surrounding a stable point. So at the stable point, we can linearize the system and kind of simplify this complex nonlinear dynamic into a more simplified linear system at the stable point. And we do that with the Jacobian. And then this Jacobian would actually be able to tell us a lot about the behavior at that specific point. So if you take the eigenvalues of this Jacobian, it will essentially tell us how that system will behave, if it's going to expand or if it's going to shrink. And in addition to that, if it's going to rotate or if it's not going to rotate, it's going to stay fixed, or like it's not going to move around. So, and that's given by the different components of the eigenvalues of the eigenvectors of the Jacobian. So it's made up of a real component, which we will call sigma over here. It's different rotations everywhere, but over here we're calling it sigma. And then we have a imaginary component, which is omega in this case. So now we're sticking with the theme of oscillations. We're trying to see if we can predict the power and the frequency of our system without necessarily having to simulate it. And so, this is the same model that you've been exposed to before. And I'm just going to show you what it looks like, what I mean by that. So for example, here, I'm getting some, what I'm doing here is I'm simulating the data, and I can see that I actually do see some oscillations. And when I take the power spectrum of it, I can see that, yep, I have a massive peak around the oscillatory frequency, and then it's subharmonics. Now, the analytical solution suggests that we can estimate the amplitude and the frequency we can make assumptions about the amplitude and frequency depending on the real part and imaginary part. So the amplitude would be scaling with the square root of the real part, whereas the frequency is the imaginary part divided by 2 pi. Now, what we're seeing here is, well, something interesting. Here, over on the left side, where we see the amplitude versus the real component, the sigma parameter, and on the right we have the angular frequency. So on the left, it kind of the blue dots here are simulations, and the red line here suggests the theory that they should kind of follow this curve. So in the case of the amplitude, we see that somewhat follows that sort of pattern of activity, but it's not necessarily the case for angular frequency, which is kind of interesting, which it raises a few questions like why that is. There's a few discussion points that I brought up for you, but to not confuse you too much, a lot of this discrepancy is because what we're trying to do with the analytical solution is to only look at the immediate region surrounding a stable point with minor perturbations to it. But for example, what we're seeing here in the case of like persistent oscillations is we don't really have a stable point. We have a limit cycle, and those two are not the same thing. So even though we can actually plug in different parameters to our equations that will give us angular frequency and power, it's not necessarily the case that at all these values we actually do get a stable point. So it might be the case that the stable point actually shifts or it deteriorates, but we can still technically generate a number out of it, but wouldn't really follow the prediction of the theory. Any questions about this? Yeah.
The eigenvalues, are we looking for, is it zero real part, positive imaginary? Is that the oscillatory behavior around the fixed point?
That's one behavior, but let's think of it this way. When it's positive, essentially what it's saying is that this Jacobian is you can think of it as like a transformation matrix. What it's gonna do, the eigenvectors are telling you that it's gonna stretch out my values. If I have a positive magnitude of my of the eigenvalue, it's gonna take a point and just gonna make it a bit larger. If it has a negative one, if it's a negative eigenvalue, it's going to make it a bit smaller. So you can think of it if the value is going to be converging or diverging towards a stable point. So if you have a real, and we're just talking about the real value over here, real part of it. So if you have a positive real part, it's going to make the system diverge, just go out, right? So you can just kind of think of it that way, if that makes sense. Now, the imaginary component, you don't really need to worry about the value of it. It's just a binary. Does it have an imaginary component or does it not have an imaginary component? If it doesn't have an imaginary component, so you would, in that case, you would get stable nodes. Fixed nodes, either stable or unstable, depending on the sign of the real value. But if you do have an imaginary component, then that's when you get some oscillations. But in the case that you said, where you have a zero real value, real component, but you do have imaginary components, what you would see is probably going to be concentric circles, concentric throughout the entire phase portrait.
I'm wondering, so does oscillatory behavior in this sense refer to a stable fixed point in the Jacobian? Because like you have like purely positive imaginary,
Yeah.
you get oscillatory behavior on the face portrait of the Jacobian, but I think I'm getting confused. So is it we're seeking out stability? So I'm trying to remember all the different combinations. I think that's like negative real eigenvalues or something usually is stability, and then does that correlate to one of these frequencies? Like when we like the stability means we're consistently getting the same kind of frequency in that case.
Not necessarily, not necessarily. And it would be more when we're doing this, we're not necessarily looking at stable points, we're looking at fixed points. So there's a kind of like a very minor, not minor, but like a very delicate difference. So fixed points can be stable or unstable. It just means that if I'm exactly at that value, I'm not going to change. But if you perturb me, if it's unstable, that's going to be like diverge. But if it's stable, it's going to converge. So, and if you want to think of the different categories of how it's going to behave, then for the real part, we have three different categories: positive, negative, or zero. But for the imaginary part of it, we only have a yes or no. Does it have one or does it not have one? And so to answer your question, it would apply to both stable and unstable node fixed points. And the fact that it would have an imaginary component would tell you that if it's oscillating or not. So it can oscillate inwards or it can oscillate outwards. Or it can just like, if it's zero, which is very rare, then it can kind of like stay in place.
Right. So then in this case, when we do the stability analysis, we're attempting to define whether or not we see oscillatory behavior, and then what of those combinations does that correspond to? I'm getting kind of myself like, is that negative, real, or
Yeah. So let's take this example, right? Let's say I'm not exactly sure if this converges to zero, but let's say this decays to zero, right? So what this would mean is that I'm kind of like starting off over here, and I'm kind of like going in spirals, and at the end I'm gonna like fall into a fixed point. So what this would tell me is that, well, the system's shrinking. So if there's in the stable point, the real part of the eigen value is going to be negative because it's making it smaller. If it's doing the opposite, if it's kind of like, doing this and then suddenly it blows up, then I would assume that I would predict that the real part of the eigenvalue is positive, right? Now, the fact that this actually is oscillating up and down, this immediately tells me that, yeah, there is definitely going to be an imaginary component to it. But it could have also been the case that it just decays, just falls into like a, I mean, I can probably like generate that case. How would we do? I'll just play around with it. Yeah, okay, over here. So here, what's this is telling me that it's kind of like falling into, kind of decaying into a stable point. So this immediately tells me that there is no imaginary component, because there is no oscillation, and then the real part of the eigenvalue is negative. Yes, negative. Otherwise, you kind of like explode like that, exponential explosion.
Okay, so then this actually would show the behavior of like taking many fixed points as over, like as it like kind of moves. Like I'm imagining more so through like a face portrait. That's why.
Right.
Like because you can actually see it diverge, or
right,
or in this case, like a spiral would show up as a wave.
Yes.
Okay.
Yeah, exactly. But every instance, every parameter set would give you either one or one set of fixed points. So the fixed points wouldn't change as we simulate through this the time.
Multiple fixed.
It might have multiple fixed points, but I think in this case it's only I don't know how many solutions it has. I think it's only one. It depends when so to calculate how many, and the way you would do that is that you can say that the fixed point is defined where either both dynamics are zero. So the rate of change of both dynamics is going to be zero, right? And when you essentially set this to zero, set these to zero, then you get two different equations that you can solve for. And you can solve for the parameters that actually would satisfy both cases. And that would give you your fixed points. Depending on how many solutions you have, you can see how many fixed points you're going to have, you know. Good question. Any other questions about any part of the other previous tutorials? Because, you know, at this point it's all coming together interestingly. All right, cool. So that was another way that we can generate oscillations. So these are kind of like somewhat disparate ways that we can actually generate oscillations so far. So we covered the Kuramoto model, which is oscillations. It's like a model that has built-in oscillations. We explored, you know, we went back to the dynamical systems and saw how we can study oscillations through that perspective. Another way to do it is actually just simulation of neural mass models. And here, we're actually going to take the algorithm. We're going to build our example around alpha waves. And the following code is borrowed from one of the PhD students in our lab. And here's the GitHub repo they can check out. Does a very good job of comprehensively investigating different neural mass models if you're interested. But essentially, what we're trying to do here is to explore alpha generation through two different theories of like, where is it coming from? One theory is saying that this alpha pattern that we see so prevalently over the brain is actually a function of the interactions on the cortex. So different cortical regions are talking to each other, and the way that they talk to each other, this feedback will generate some alpha oscillations. Another theory is to say that, well, this interaction, this frequency, this alpha frequency that we see disproportionately compared to other frequency bands, is actually coming from not the cortex itself, but like the interactions between the cortex and the thalamus, different deeper regions of the brain. So over here, so like the thalamus is deeper into the within the brain, and it has connections on each of the regions of the cortex. It's a relay nucleus. And so both theories can make assumptions of how this actually can happen, and we can actually test those assumptions to see if we can actually generate alpha oscillations based on the assumptions that we make. So is that clear? Is that clear what's happening here? Okay. So here, this is the first model. This is the Yancey-Ritt equations. It's somewhat similar to the Wilson-Cowan in the sense that it's a neural mass model, but what it's made up of is three different populations that are talking to each other. Excitatory, so we have the main pyramidal cells, an excited interneuron population, and an inhibitory interneuron population. And this the connection and the communications within these different subpopulations that can actually give you that sort of alpha regime. So this is I won't go too much into the structure of the model. You can study up on it on your own, but essentially each population has a rate to potential operator and a potential to rate operator. If you remember from, I think it was lecture 2, you have firing rates turning into voltage, and then voltage turning back into firing rate. So it's kind of like following the same general principles, albeit with different transfer functions and different specifics. So here, what we're doing is that we're actually playing around with different parameters of this model. So we have, like, for example, connectivity strength between the excitatory population and the pyramidal populations, or the inhibitory the pyramidal population to the inhibitory population. So these are characterized by C1, 2, 3, 4, etc. We also have time delays, or rather decay rates of different for the different operators that turn the voltage into rates, or rather firing rates into voltage. So here, this is a intricate balance that Cerns has found, or others have found, that would give us a main oscillatory activity around the same frequency as alpha. And so this is saying that if we is through our model assumes here that, you know, this is all happening within the cortex. So it's cortical interactions, and through the cortical interactions, then this phenomenon of alpha can emerge. This is one model. The other one that we can look at is the Robinson-Rennie-Wright model, which is a bit more complicated, and it involves the thalamus as well. So the thalamus has two different subpopulations, the relay nuclei and the reticular nucleus. And the cortex talks to these regions either directly or indirectly. And so the assumption here in this model is that this alpha is actually happening through the time delay between the cortex and the thalamus. So kind of like, you know, there's a time delay that's happening for the communication, and that would actually give us that kind of like feedback interaction, which will give us that sort of oscillatory behavior. So it's the same principle here, that, you know, again, I advise to go over the different parameters to understand what they mean. There's a little bit of a synopsis over here. But if we take that assumption, we can also see that, yeah, that this type of interaction can also give us an alpha generating regime. So these are coming from two different assumptions, underlying assumptions, a cortical origin or a corticothalamic origin of alpha, but we can generate alpha oscillations from both of them. So what your exercise, there's an exercise for you guys, which is relating these neural mass models to different modes of behavior. In this case, eyes open and eyes closed. So as Dr. Griffiths was mentioning, in the case that we have, when the eyes are closed, we see this sort of alpha bump over here, where we have increased alpha activity. But when the eyes are open, this is at the back of the head, at the occipital cortex. When the eyes are open, then we see this bump decay. So you can actually see this, there's a predominant alpha activity going on, but when the eyes are opened, then this goes down, this decays. So your task here is to actually use the widgets for each of the two models to see if you can replicate the two different instances of eyes closed and eyes open, and see what parameter is it that you're changing, what changes are you making to the system that's actually making this transition happen. And to infer, well, does that make sense? You know, would it make sense for like alpha to, if for example, I see that the connections between the inhibitory and excitatory change when I open my eyes, does that make sense? Would that be a sufficient explanation of me opening my eyes? Or would it depend, for example, on the input or the noise input to the system? So this is kind of like a bit of a parameter exploration for you guys. So it's laid out here what you guys need to do. And this is actually a good example for the presentation at the end of the course that you guys have to do. So a little bit, just a bit more detail on the presentation. It's going to be about 7 minutes of presentation on one of these exercises throughout the notebook that you find interesting. So you have options here. But, you know, again, you have to talk about it for like 7 minutes. So, you know, I suggest you pick one of the more interesting, more elaborate ones. But essentially, the way that it was going to go is 7 minutes of presentation over the code, and you don't need to prepare like if you don't want to. You don't need to prepare PowerPoint slides or anything. You could go over the code and just explain well what this interaction is saying and how you interpret it, and just talk about it to give the sense of that you actually understand what's going on here. So for example, an instance of it is in this example, I vary in the Robinson model, I vary the input, the relay nucleus, the input from the thalamus to the cortex. And I see that when I increase it, the alpha goes down and it replicates the eyes open condition. And I can say, for example, well, that makes sense, or this would make sense because the thalamus is acting as a relay nucleus. So when I actually open my eyes, that means that there's more signal coming from the thalamus to the occipital cortex. And when that happens, I see the alpha go down. So, you know, this is just an example of what you can talk about. And or you, for example, you can talk about the code or about how these different models are structured, what different populations are they're made of. But essentially, it's going to be the final lecture, or yeah, in the final lecture, it's about 7 minutes of presentation for each person, and 3 minutes of Q&A. So that was a kind of like a big parenthesis in the middle. Any questions about that? April 16th, I believe. Yeah, it's on the course website. Yeah, April 16th. Yep. Okay, let's see. Yep. So that was a lecture. That was a lecture. So we covered oscillations in different perspectives to study oscillations, the Kuromoto model, the dynamical systems perspective, stability analysis again, and finally more phenomenological approximations using neural mass models at different scales. Any questions? Yep.
So like when we started the tutorial, we kind of briefly showed that there's a whole bunch of different frequency ranges. But
Mhm.
is the popularity of alpha frequency specifically because it just is like so prevalent? And is that also why at the end of the tutorial you kind of show different phenomenological reasons as to like why do we keep seeing this frequency show up so often?
Right.
And also to like further note on that, like very low level thought process, wouldn't you maybe think if you have one reason for one frequency to exist, shouldn't that reason extend for every other frequency? In a very like rudimentary basic kind of thought process, because I was like, you were showing different ways, different parts of the brain, how it could why it could logically generate this alpha frequency. And yeah, is that like a common practice to kind of segregate the different frequencies to different logical reasons, brain areas and whatnot? Like yeah. And overall, just like the whole,
Yeah.
you know, we've talked a lot about alpha also
Yeah.
again as well.
Like what's up with alpha?
why
Why is it so special?
should be
Yeah.
why specifically this one?
Yeah.
Different one? Yeah.
That's a very good question, and I feel like John would have a lot to say about that, especially since we study alpha.
I want to say, I think I've said it several times today, but it's exactly the right question.
Yeah.
But although the way in which that's the case is that we often talk about the models. I mean, all the models that we've talked about today as like model of alpha, model of gamma, so on. And so sometimes they are exactly that, right? Oh, but other times, and so I think for the ones that we've got like on the slide, for example, these alpha models, like they're not just models of alpha, they're models of the power spectrum, right? But it's a power spectrum that has certain features, of which the two principal ones are a dominant frequency at alpha and one at other end. So within that kind of framework, you know, you can add some stuff in. You could have the easy thing to add in is like harmonics, right? So you could have a beta frequency component at a harmonic of alpha, and that kind of would come out of that kind of system quite easily with certain configurations for parameters. Adding in like another frequency that's not coming directly out of that circuit, maybe you have to have another like circuit element to get multiple frequencies. So like but you don't necessarily want multiple frequencies. So if you just at the kind of macro level of most of the data that we see in human brains and human EEG and so on, you know, there isn't much more than that. Like the alpha, one over F, a little bit of beta, that's what's there. Now the trick is there's other frequencies and oscillatory kind of phenomena that you measure different modalities, right? And different scales. So this is like, do you have like a one size all model? No, because there aren't any one size all models that work. If I have a model of alpha that doesn't really generate any gamma, in terms of like a peak and gamma, as opposed to just like the broadband, you know, one over gamma range. Is that missing something because it doesn't have gamma in it, right? So that would be the kind of philosophical question behind that. But I think the philosophical question behind what your intuition there was, I entirely agree with. It's like you want to have something that spans the range, and in some sense, like generates all frequencies. It's holistic in the sense of it is a model of the spectrum. It's the spectrum over like a 1 to 100 hertz range or something like that. Then yeah, but the question is like if you want to account for more, what is the data and the kind of phenomena that you're specifically talking about if you were to have more than just that? And what would be the rationale for having a kind of larger model or a more complex, you know, set of equations that has extra things in over and above at least a kind of simple holistic view of, you know, 1 to 100 hertz. But yeah, so they're not just models of alpha, they're models of the power spectrum with the dominant frequency at alpha. It's
Is there any like, I don't know, like hand wavy philosophical kind of interpretation for why we see it so much? Like, do we see it when we sleep? Do we see it in like, I don't know, like fetuses or something, like in this kind of idling state?
why is it so dominant?
Yeah.
I mean, I don't know to what extent this is, you know, a rigorous explanation, but could it be thought of as like a clock cycle of the brain, of different, because we talked about the gamma and alpha phase coupling, you know, and then you have like, you know, more refined computations happening at the specific phases of alpha. And there's kind of like a depolarization wave that's coming in. And at some point, you know, whatever values are close to being fired, then you get that kind of like push to either fire or not. And then you kind of have like that kind of like CPU type clock cycle, if that makes sense. But I don't know.
Yeah, I mean, if you want to go there, then that works, and that's like a live idea. I was trying to get at that in some of the things or something like in the slides. I think there's a related thing about how, well, yes, a clock cycle kind of makes sense in terms of, particularly if you think of the kind of gamma and the firing, you know, as being kind of a pair of things that go on they're fast things, right? So the reason why alpha would make sense as a kind of a bit of a clock cycle is because that would be a structure that you can collect several fast things on top of in a slow frequency like basket, right? So yeah, there's like binding type things about what's the functional role there. But I think the original question of why, no one knows. Like that is the question, really. I have this this little kind of hobby horse that I've been jumping on for a while because we're at like 100 years of alpha, right? I took that burger, didn't mention that I took that burger. That was the paper that's the citation is 1929, but the original work was done in 1924. So we're actually like just we're in the centenary of alpha, and we don't know where alpha comes from in terms of having solved the question of what's the correct model, and that's a bit galling. I mean, that's kind of embarrassing in a way, but it's also, I think, exciting from a scientific point of view. It's like an unsolved problem. It should be a solvable problem, but it's an unsolved problem, and it is the biggest thing you see in the brain. It's the most dominant, you know, property of measurable human brain activity, but we don't know the answer to your question why yet.
Do less complicated nervous systems still have this?
Yeah, well, that's another good question. So yes, mice have alpha. I believe it's the case. So there's a story. I think I just have to say it's an anecdote, or at least I don't know the full accuracy of this, but after Berger discovered alpha and discovered its kind of dependence on isochronous closed in the end of the 20s, then there was like skepticism broadly and excitement. And then about 5-10 years later, there was a bunch of dudes in Cambridge in the UK. They kind of consolidated the uptake of Berger's work by doing two things primarily. They improved some of the reporting techniques because they were slightly better kind of engineers in terms of doing experimental electrophysiology. And they also did it in other animals, including flies. So I think the story that I don't know is what the truth of it is. I tried to look it up. So what the is that something like, so the guy who led this, his name was Adrian, and he had a Nobel Prize for some of the other stuff. So he was like a famous scientist, and what Berger was a famous scientist. Berger was something like the back. It's a very good point of view of clinical science. So Adrian kind of put Berger on the map basically, but at the same time, he sort of dismissed the interest, the value of the kind of observation of this rhythm and its dependence, but because they said, well, you can see it flies, so it's not that interesting. It's flies out, that's fine. Now that's wrong in lots of ways, but I guess you can see where it comes from. So that's a long-winded way to a little bit cheap anecdote saying, yeah, you definitely have like algorithms and the analogs of algorithms across a range of species going back a long way. But the flies thing, I'll look into it. So it's possible flies also have algorithms, which is obviously important for a question of like what's the mechanism, what's the circuit, right?
Yeah, exactly.
So it's always taken to get the answer.
I wonder if it's maybe been thought of kind of like maybe like a law of large numbers kind of way, like a central limit theorem, where it's like if you involve all these different processes, you just happen to get this wave that is just the middle of the Gaussian kind of thing. Because you said like you equally weighted all of them, all 10, but yeah, but alpha shows up a lot. In that kind of way, like maybe it's because you see throughout like physics and life, you can see a lot of central limit theorem, bell curve-esque things. Has it been crudely thought of maybe in that way?
Well, there's actually a thing related to that Paz and I were talking about in the week coming up to this, which is there's some work from actually a colleague of mine that had this kind of idea that for the Kuramoto model, you can get the emergence of low frequencies from a network of high frequencies in those systems, and that's a bit like one version of what you're getting at there, which is like have a bunch of high frequency oscillators. Then you can get a kind of almost like a beat frequency in which they will synchronize around. There's a lot of different frequencies. So I knew about this from kind of like high level knowledge of this work for a while, and then I looked into it. I was like, we couldn't get our head around what the mathematical description of that is. So I'm not after having basically taken a closer look, I'm not really sure yet, but I can point to the references on that. But there is like at least an idea within the context of Kuramoto models about like emergence of low frequencies, and that's kind of part of that. The things you're thinking there. Yeah. It's slaving. It's synergetics. Slaving.
Entrainment?
No, it's like that's the word. It's called. So it's like you have a system with fast timescales and slow timescales, and the like enslavement principle from Hartman from the 80s is like that the slow time scales will take over, and the fast time scale components will kind of lock into the slow time scale process. And so that's, I think there's a bunch of systems that they've studied that around lasers as well, but we don't know how it works with lasers.
I have heard about that with like colors, like the good basic examples, like you just mix all the colors, you get like a weird brown kind of thing. But it's like you could also view it in like the waves, the actual color waves, then you kind of maybe extend it to like the outright picture output. It's showing up.
But the average, I mean, the thing about it being the average, like the kind of the opposite of that, about the 1 over F, right?
Yeah.
I mean, the alpha coming up in the middle, maybe, but the general idea of that is that it's, you know,
Scale invariance.
scale invariant. So,
I mean, you see that on, like in many different systems, all the systems around you, and in the complex system that's stable, you see that kind of like 1 over F component that's just prevalent, including the human brain. It's just that the human brain also has that alpha peak, which I don't think is necessarily present in other systems, which is interesting. And then, you know, another question is like, why, in that case, why would it be alpha if there's a scale invariance? You know, there's a is it the average? Is it like the special thing about alpha? Is it the average of all the different frequencies? But you know, then again, you can just extend the different frequencies to like, you know, however large.
it probably is the one in the middle. Think about like the relevant frequencies in the brain. It goes from like infra slow, which is like 0.01 hertz, to about 100 hertz. And 10 hertz, it's almost that. But maybe check back in 5-10 years and we'll solve that. You know, 105-year anniversary, and let's give a clear answer to the question. This course. Thanks, round of applause to Plaza.
Thank you very much.
Tutorial. Yeah, I hope everyone was clear on the recommendation of the structure of the assignments that we'll be having from you on the last day. You know, to summarize his point, just a 7-minute presentation following one of the ideas that are offered there's lots of options for you to choose, you know, from the notebooks that you've had. If you want to pick a new one, okay, but we would recommend you to just take one of the kind of written questions in notebooks, and then like literally what you're going to do is just take the notebook, copy it, maybe get rid of the earlier stuff, although you don't have to, but then just talk us through what you add like narrative content, code, images, and just stand up and share a screen in the way that he's been doing it. So you don't need to prepare a slideshow, you don't need to kind of like waste time on PowerPoint, just do it in a notebook and then expect to be, you know, scrolling through the notebook and talking through your stuff that way. Okay. Great. Have a good day, guys.
Take care,
Have a good
everyone.
week. See you next time.